Okay, so this is continuing category 2A, assemblies of molecules, cells, and groups of cells within single cellular and multicellular organisms. So we've been kind of looking at it from more of a biochemistry aspect. Now we're going to look at it straight biology. We talked about lipids and we talked about, how they play a role in membranes. So we talked about these phospholipids, right? These and their structure where they have these two long fatty acids and then a negatively charged phosphate head group. So we talked about how the top part is hydrophilic, right? And it interacts with the water. The bottom parts are hydrophobic and they interact with themselves, right? So we can see the right here that this, let me draw it in blue so it makes sense. So this, the top, the heads, the bulbous heads all up here, all interact with the water, right? And then in yellow, the hydrophobic tails, right? They just stick to themselves. And that gives us a nice, a favorable kind of conformation. So now we'll talk more about how, yeah, that's that. And we talked about factors that affect the structural integrity of the membrane, the rigidity of the membrane, right? So we talked about if a fatty acid is, if the tails are longer, right, they can pack together more tightly, and that makes the membrane more resistant to temperature and stuff like that. So now we'll talk about, so that was a deep dive into lipids last week. So now we'll talk more about other components of the membrane. So one of the major things that we were talking about before was how does stuff get around the membrane, right? Because we have these properties of hydrophobic and hydrophilic, right? So what if something hydrophilic wants to get through this hydrophobic layer, right? In that case, we need to have some kind of transport, some way to transport that stuff, right? And that's where we have channels and pumps, right? So if it's so let's see here. We talked about the different types of diffusion stuff. Oh, let me open this up. Okay. Oh, no, I need to share this. Can you see that I changed the screen? Did I do that properly? Is it still stuck on the web page? Maybe I need to
No, it's on the Anki.
Oh, okay, good. So yeah, we were just going to talk about the different types of transport, right? So we're talking about, we have this membrane, right? And how do we get stuff through the membrane? Well, if it really depends on the molecule that's going through the membrane. If the molecule is small and non-polar, right? It can directly go through. So we're saying, remember, why does it have to be non-polar? Because the middle part of that whole region is non-polar. And I'm going to try to use like yellow to show non-polar and blue to show polar. So remember, this whole region is non-polar. It's hydrophobic, right? So these guys, if the solute themselves are non-polar, then they can go straight through because they're okay to be in this environment, in this nonpolar hydrophobic environment. Now, if the molecule is polar, right? If it's a polar molecule, then it's going to have a hard time being here, right? If it's a polar molecule, it doesn't want to be in this nonpolar environment. So it cannot go straight through. So what is our solution to that? Well, we can make a channel for them to go through, right? And now they're okay because this protein is probably like negatively charged or charged in some way, right? Polar just means like charged, and hydrophobic kind of means like uncharged, right? So these guys, the inside of this channel is probably charged to make these polar molecules happier to go through this way, or more stable when going through this way. So notice that in both cases, we are doing passive transport, because passive transport means high to low concentration. So there's more of this stuff here than there is here, all right? So when we go from high to low concentration, we're dealing with passive transport. Now, the reason that these is two different boxes is because one of them is called simple and one of them is called facilitated, right? And simple means it doesn't need anything fancy. It can just go through straight, right? And the reason it can go through straight is because those molecules are nonpolar and they match the inside of the bilayer, which is also nonpolar. Now, if it doesn't match, if the molecules are polar or if they're large, right? Also, large molecules may have a hard time going through the membrane directly, right? So if they're large or polar, then they would rather go through a channel like this. And this kind of channel can facilitate or help, right? So this is a facilitated transport. But the key thing here is they're both passive transports, because the word passive means that there's no energy being expended for this to go. If they go from high to low, it doesn't cost energy. And that's the main thing. So that's in contrast to active transport, right? So if passive transport doesn't use any energy, then active transport uses energy. And for active transport to use energy, this is kind of what it looks like. So the simple one is that, so again, notice how in passive we go from high to low, right? But in active, we're going to go from low to high. That's what makes it active, right? We're pushing normally, like all molecules and stuff go from high concentration to low concentration. We call that a concentration gradient, right? But if we go the opposite way, if we go from low concentration to high concentration, then it's going to require energy for that to do. Any molecule trying to go through that's charged, yeah, it doesn't even have to be fully charged necessarily. Like there are polar, if a like for example, if it just has like an OH on it, you can consider it polar because it has some polarity, because O is electronegative. It doesn't have to be full on charged. Things that are hydrophobic are like just carbons and hydrogens because they have no charge and they have no electronegativity either. But molecules that have oxygen in it, even if it's not fully charged, they can still be considered polar, because that's more along the lines of like organic chemistry. But electronegative molecules, like the ones at the top right of the periodic table, like chlorine, bromine, oxygen, those guys have a tendency to pull electrons. So when they pull electrons, what they're doing is they're taking it away from their neighbors. So to just draw it simply, if we have a carbon attached to a carbon, this is completely equal sharing of electrons because the electrons don't prefer either atom. But then if we have a carbon attached to an oxygen, this is not even sharing because the oxygen wants the electrons more than the carbons. So when that happens, we can draw an arrow like this. And that arrow represents that this is a polar bond because the electrons are going to want to be on this side more. The electrons are more distributed towards this way rather than in this case where the electrons might be like even. So that's essentially what polarity means. It doesn't need to be that this oxygen is negatively charged. Even if this oxygen is just an OH, that oxygen is still electronegative and it's still kind of sucking away the electrons from its neighbors. So that's what polarity is. Okay, so we were talking about active transport. So active transport is the solutes are transported across the membrane using energy from ATP hydrolysis. So what that means is ATP molecule is here. It is hydrolyzed, so it's cleaved, and it turns into ADP and a phosphate, right? So ATP is triphosphate, ADP is diphosphate, plus another phosphate. So that's our 3 phosphates. And when we cleave that bond, it's a high energy bond, and it provides us the energy to do some action, right? And in this case, the action is moving a molecule from low to high, right? So this is a form of active transport because we're using ATP, and it's direct or primary active transport because we used it for the express purpose of moving something, against its concentration. Now, there's another form called secondary active transport, and secondary active transport looks like this. So in this case, we saw on the left the primary active transport, right? What we did here was we pushed sodium from its low to its high, right? Because there's only 3 sodium here, but there's like 5 sodium up here. So we pushed sodium to its high, right? Then, because the sodium up here is now higher, right? The concentration of sodium up here is high now. Let me just write this down. So the concentration of sodium, high sodium here, right? And the concentration of sodium below is low sodium, right? So what is the sodium going to want to do? It's going to want to go from high to low, right? And that's what this yellow arrow is showing. The sodium is going to want to go back down, right? However, at the same time that it goes back down, we can accomplish some work with this. And we do this through the form of a co-transporter, right? Means it's co-transport. So it's going to transport 2 things at the same time. So what's happening is as the sodium comes back down to where it wants to go, a glucose is going to be brought against this concentration gradient. Because we can see here the glucose is actually low glucose here in the top or extracellularly, and then it's high glucose below. So we actually, again, did another form of active transport right here with the glucose. So in the first one, in the primary, we performed active transport on the sodium. In the second one, we performed active transport on the glucose, right? And the main thing to see here is that we only broke 1 ATP, only when we did the primary movement, right? So why is the second one still counted as active transport? Because we used the ATP to move the sodium up there in the first place. So the effect of ATP was still happening. This secondary doesn't happen if we didn't use ATP, right? If we didn't use the ATP to move the Na up, then there wouldn't be any Na to come back down. So because we had to use an ATP for this transporter to have any sodium in the first place, this is considered secondary active transport because we use some ATP to transport the sodium across the membrane, and then the energy that's released by it coming back down is harnessed to move something else. So this is the 2 types. Okay, so any questions about these 2 types of transport? The big categories are passive and active, and then simple versus facilitated, and primary versus secondary. Okay, cool. So membrane transport, pretty important. Now we can move on to, so we talked about membrane channels. Oh, no, we talked about solute transport, but membrane channels is kind of a slightly different category. We also have this from regular bio, this idea of isotonic, hypotonic, and hypertonic. And this is just what we saw with diffusion, but diffusion of water. So in this case, right, if the water will go to wherever there's more solutes, these gray dots. So the water is inside the cell right now, right? If there is more, so they're not showing you, but there's more dots inside the cell compared to the outside here. I don't think I can draw enough dots, but the idea is that there's more dots in here. And because there's more solutes in here, the water is going to come down inside the cell. Whereas in this picture, it's probably easier to see, there's more solutes on the outside of the cell. So the water goes to wherever the solutes are. So the water leaves the cell and goes outside. So, and there's less water left inside. Okay, so we talked about transport and all of that. So now, the sodium potassium pump. Yeah. So this is a pretty important thing. You're going to see it more when we talk about active not what's it called? It's not action potential, yeah, and neurology, yeah. So that's important. So the sodium potassium pump is basically 3 sodium come in and it uses ATP. So again, this is active transport. It uses 1 ATP to move 3 sodium out of the cell, 2 potassium into the cell for the cost of 1 ATP. So this is gonna, I think it's right about to come up because I think we're gonna talk about membrane potential soon. And there's also, so we talked about, this is called a channel protein because it's not a pump. A pump and a channel is different. A channel is just helping, it's just facilitating, right? It's not using any ATP to move anything to its high concentration. This is just allowing things to flow down their correct concentration, but it's providing a path through the hydrophobic environment, right? So again, this bilayer right here is hydrophobic, but the protein channel here, this is not a good color, but the protein channel here is hydrophilic. So it's providing an alternate route for the stuff to go through, but it is not involving any energy. It's not using any energy to allow things to go through. If it was using energy to allow things to go through, then we call that a pump, not a channel. Okay. And one of the important ones is probably aquaporin, right? So aqua, water, porin, a pore, right? So it's a pore through the membrane that allows water to go in. And for certain cells, that's important to have like a easy access of water. And the channels are not necessarily always open. They can be gated. So what it means if it's gated is that it can be a gated channel means that it's open, but only under certain conditions. So channels. No, this is not. So basically, the basic idea is that channels can be opened and closed depending on various properties. So if it can be ligand gated. A ligand means that some molecule has to attach for that thing to open in the first place. It can be voltage gated, means that the membrane has to have some voltage for it to open. And you're going to see that with action potential. So I think we're going to talk about membrane potential right now. Let's see. Yeah. Okay. So this is a pretty interesting and in-depth topic. So we're going to try to break it down a little bit at a time. So we know that we have electricity in our bodies, right? We know we have electricity in our brain. We know we can measure the electricity in our heart, right? How does that actually work, right? So how that actually works is based on very interesting dynamic between charged ions that we have, right? So if you measure a membrane's potential, right, you will see that it is a little negatively charged. Usually, yeah, resting membrane potential is between -30 to -90. I've usually seen it as like -70. Most people just say -70 millivolts, right? So what is that? What's causing that? What's causing that is because of the environment of ions that we see um, at normal conditions, right? So in normal conditions, we would have more potassium inside and more potassium outside. The reason that this is happening is because, wait, did I say that right? I said more potassium inside and more sodium outside. And the reason that's happening is because of this pump. This pump is maintaining this scenario. The pump is constantly feeding potassium inside the cell and constantly forcing sodium outside of the cell. And that's maintaining this -70. Why is it -70? This is not exactly the reason, but this is a good way to start thinking about it. The reason that the cell is negative, right, is because it's pumping. Remember, it's not the same because it's pumping 3 sodium out and 2 potassium in. So in a sense, you can think of it as if 3 positive is going out and 2 positive is coming in, then that's kind of like a net of the cell. The cell lost 1 positive charge. You see what I'm saying? Like if 3 positively charged sodium comes out and 2 positively charged potassium comes in, then overall the cell lost a positive, right? So if the cell lost a positive, and this is happening a bunch of times, then this accumulates into an overall slightly negative charge of the membrane. So because with each transaction, the cell is basically getting a -1 charge. And so repeat constantly, and then you eventually get a resting membrane potential of -70 millivolts. So this is not the exact explanation, but the exact explanation is incredibly complicated and beyond the scope. So this is just a good way to start thinking about why it's negative at when it's normal, right? Like a misconception would be to think like, oh, nothing is happening, so it's probably 0 millivolts. No, it's always -70 millivolts at resting. And so now this is just resting, but other things can happen. Other things can happen to the cell. So remember that we talked about polarity, right? We talked about polar molecules, right? And we said that polar molecules are negative, not necessarily negatively charged, but they have negativity, they have more electrons, right? So in that sense, because this membrane has a negative charge, right, a negative charge of -70 millivolts, or a negative potential rather, then we can consider the membrane polarized, right? If it was 0, it would be nonpolar. There would be no difference, but there is a polarity here, and the polarity is right over here. I can show you. If we had to draw out like a polarity arrow, then we can see the polarity is like this, because the inside of the cell has less positivity than the outside of the cell. So the arrow shows kind of, wait, did I draw this in the right way? Wait, I think this is the positive end. Whoa, hold on. Dipole arrow. Hold on. Let me see if I remember what a dipole arrow looks like. Dipole arrow. Dipole arrow points towards a more electronegative element. Oh no, so then I think I drew it in the wrong way. So then the dipole arrow, because the more electronegative element is the inside of the cell, right? Because there's less positive stuff in there. So then it should be like this. Oops. Because because this side represents the positive. And then but okay, you don't need to worry about that really. But basically, the membrane is considered polarized. It's considered polar because it's at a negative charge. Now, the membrane can become depolarized. What that means is that it was originally negative, polarized. So to become depolarized, it becomes positive, right? It becomes neutral. So when it becomes neutral, it is depolar. We consider it depolarized. It was originally polarized, now it became depolarized. How does it become depolarized? Well, originally the situation is we are dumping the sodium out and we're letting the potassium in, right? So we said we are losing some positive charges from the cell. So if we're losing positive charges, from the cell, losing positive charges from the cell leads to polarization. So therefore, doing what would lead to depolarization? If losing positive charges from the cell leads to polarization, then blank positive charges to the cell, right, leads to depolarization. So gaining positive charges to the cell leads to depolarization. So, and that's accomplished through what we call a sodium channel. And a sodium channel is just allowing a straight up the sodium to come back in that we pumped out. So when the sodium comes back in, right, it is giving the inside of the cell more positive charge. If it's giving the inside of the cell more positive charge, this charge is going to go from -70 back up to 0. So that is how, so it's really just tracking the sodium and potassiums. That is why we have a charge, right? Now, the interesting thing is when the sodium channel is a little slow to close. So or is it the potassium channel? Well, anyway, the sodium channel, when it comes in, the sodium will keep coming in. So it won't just stop when it hits 0. The sodiums will keep coming in. So when they keep coming in, we were polarized, then we became depolarized. Now we passed depolarized and we became hyperpolarized because the sodiums come in more and more and more, and it actually flips it and the membrane potential is now positive. And when that happens, the cell will open these K+ leak channels to get the potassium out, because this is another way to dump the potassium, to dump the positive charge. So once we dump the positive charge, eventually it will allow the membrane to kind of reset and go back to this state. So this is overall like the very small, this is on a very small scale what's happening with action potential. And it's going to be hard to understand until you see all the pieces of how an action potential works. Yes, this is really what we just saw was how the action potential works on the membrane level. So let me just show you action potential. So we've seen this kind of, you've probably seen this kind of graph before, where it's talking about you can see membrane potential on the left, right? It's talking about how a membrane experiences action potential. And how that happens is through what I've kind of shown you right now. So the first step was the resting phase, right? And where is the resting? At -70. So that's the membrane potential, right? And what's happening there is, it's not shown here, but the potassium pump is pumping sodium up and potassium in, right? Then when we have depolarization, the sodium channels open, and that's what I showed you in that picture. When we have depolarization, so this is resting at -70, and then at depolarization, the sodium is allowed to come in. So at depolarization, the sodium is allowed to come in. And because the sodium comes in, the membrane potential increases and it becomes positive, right? Then we have, okay, repolarization. Yeah. So the repolarization is when the sodium channels have closed and the potassium channels open, right? So we can see that here. The sodium channel is closed and the potassium channel is open and the potassium is going out, right? So that's allowing us to come well, the kind of depolarization was here at 0, right? But then when it increases, it kind of becomes, oh, they're considering hyperpolarized that way. Oh, no. That's not really correct. This is depolarized. This is still depolarized. This is all depolarized. This is depolarized because depolarized means that you're going more and more positive, right? Depolarized. Polarized is it was negative. It had electrons. Depolarized is becoming more positive. So all of this is kind of depolarizing. And now in this part, we call it repolarizing because we're going back to -70. We're trying to go back to negative. So we're repolarizing. And then here, what happens is we become hyperpolarized because we go past -70. We go to -80, -90. Why? Because these potassium channels are slow to close. And so that happens. And then eventually we return to resting membrane potential, which is at -70. And so what the pictures were showing you before was kind of how the sodium and potassium plays a role in that, because it's really all caused by the movement of these positively charged ions. So that's kind of, so this is how it's all linked together, right? We talked about those channels, we talked about the pump, and now that knowledge is going to help you understand better how an action potential works and why it works. But this is we'll come back to this again when we talk about nerves and the nervous system. It's going to be a huge thing. But for now, you have a little bit of an exposure to it. Okay. Yes. And so, oh yeah. So we were talking about the different. Okay. Any questions about action? Don't worry if you don't like get it right now because we're going to go into it like more in depth. Like I said, when we talk about the nervous system, when we actually break down the whole everything about how a neuronal impulse actually works. So yeah, so we were talking about, so we talked about channels and pumps, but how do they actually are regulated, right? So I said there are ligand gated receptors or channels, and a ligand gated thing is closed unless the ligand is bound. So for example, here's an example of a neurotransmitter, right? Maybe you don't want to accept sodium unless the neurotransmitter is trying to get a signal across, right? And then if the neurotransmitter binds, then this thing opens and sodium is allowed to come in. Hey, what happens when sodium is allowed to come in? An action potential, right? So we can start seeing how these things are all kind of linked together. You just need the building block one at a time to get the full picture. So we can see here a neurotransmitter is binding and a neurotransmitter is allowing sodium to come in. And that is how a neurotransmitter can start an action potential, can start a response through the cells, right? So just a little bit at a time, baby steps. And notice how the sodium cannot get in if the ligand is not bound. So this is one type of regulation. Another type of regulation is mechanical gating. So mechanical in this sense means like physical. Like for example, if you have pressure if you put pressure on your tissue or something, or there's even like in your ear, there's you know how like when you spin around you get dizzy? It's because there's fluid in your ear, and that fluid can kind of push on hairs in your ear to open certain channels. I think it's actually it's pretty cool. Let me see if I can find it. Kinocilia. Yeah. So here you can see we have these like little hairs on the inside of our ear, and when that gets moved, it triggers some stuff. It triggers like movements inside of proteins and stuff. Yeah, but we'll get to that. Don't worry. We have anatomy to come. Oh, here's a good picture. When stereocilia bend, a sequence of events results in the release of neurotransmitter. So see, the question is, what is triggering the chemical signaling, right? Here, it's not a chemical that comes and attaches. It's just that there's pressure from the liquid in your ear moving. And when that pressure happens, we get some depolarization. So that pressure gets converted into chemical signaling, right? The pressure actually turned into chemical signals inside the cells. Whereas before, this is still a chemical to chemical because a neurotransmitter is a chemical, right? So we can highlight like what is actually causing this. In this case, in ligand gated, it's a neurotransmitter, which is a chemical. In mechanically gated, it's pressure, which is not a chemical, but the pressure can get turned into a chemical signaling. Then we also have, obviously we have always open, like not gated at all. If it's not gated, then it's just always open, right? And that's usually for like leak. Those are usually called like leak channels because they're constantly leaking whatever the solute is out or in. And last one is voltage gated, right? So this is very important. Again, you're going to see it with action potential. Voltage gated is responding to the charge on the membrane. So for example, if we go back here, this sodium channel is voltage gated because while the sodium potassium pump is running, this channel is closed, right? It's not just taking the sodium back inside the cell. The pump pumps it out. The sodium channel is not doing anything, right? However, once we reach a certain threshold, right? So what would happen is that there would be electrical signaling coming from another cell or something, and that would bump this up a little bit. And if we look at the graph, you can see what I mean. You can see that there is a little bit of an increase, right? A little bit of an increase until we hit what's called threshold. So when that little bit of an increase hits threshold at -55, that's the trigger. That's the trigger for the voltage gated sodium channels to say, okay, this is an action potential. Let me open and let me literally open the floodgates and allow the sodium to come in. So that's what we call voltage gated. It responds to the voltage of the membrane. And so when there's a change in the membrane potential, it triggers a difference in the open or closing of the voltage gated channels. Yeah, voltage gated. Yeah, that's so.
Really never, I was like, why has that just hit me right now this whole time? I remember being in like mammalian physiology, hearing these terms and never thinking about it like a gate, and that the term behind it is the thing that activates the gate.
Yeah, that's
It's way simpler thinking about this. I just never thought about it this way before.
that's the fun part of this for me because it's like there's so much stuff that like you just didn't have the time to make the connection or you weren't taught it in a way that like made the connection. But then once you sit down and you're basically going back through everything that you've already learned, right? And you're gonna make new connections and new like, oh, that makes so much sense now. Like that was really the case for me, especially in undergrad. Like I had a really hard time with like intro bio, and a lot of the stuff only made sense to me when I went back and I had like a chemistry background. That's why I talk in terms of like polarity and like stuff like that, because like the chemistry makes more sense to me than the biology. But that's a little bit of tangent. Anyways, yeah, that's good. That's great to find those little moments of like clarity. It's good. So what else? Yeah, so this is again another type of ligand gated, right? Like ions can attach or signaling molecules can attach. Um, oh no, sorry, this is not what that's showing. This is just showing a ligand gated. The signaling molecule attaches and the ions are allowed to go through. We talked about G protein linked receptors in the past. Yeah, they're basically we have 7 transmembrane domains and they allow for channel signaling. That's okay. So any questions about all that stuff? All of that stuff. So so now we're gonna go more into like the cell, right? We've talked a lot about the membrane. Now we're gonna talk like more about the cell and like things that happen in the cell, right? So you may have heard of so endocytosis, right? Is This is still along the lines of like moving stuff into the cell, but in a slightly different way. If it's a big thing, right? If it's a big thing and there's not necessarily a channel or a protein that allows for it to go through that's specific for it, then what the cell can do is actually just make a vesicle and just bring the vesicle in, right? So it's it is active transport, right? Because you have to move the large molecule around, and the plasma membrane will kind of pinch off, like so it's gonna like right here, it's gonna pinch off, and that's what's gonna become the thing that goes inside. So depending on what we're bringing in, we call it different stuff. It's called phagocytosis if we're eating like food, if we're eating like random eating is like a weird term, but it is eating, but it's basically like taking something in for breakdown, right? That's kind of what we mean by phagocytosis. So large particles like cells or viral particles are taken in, and then we go to a lysosome, which allows to break it down, right? We'll talk more about what lysosomes are. Penocytosis is cell drinking. So phagocytosis is cell eating, penocytosis is cell drinking, and basically uh, we just take in water molecules through this way. Um, the Originally they thought that what like we had to take in water through like this, but then we learned about aquaporins and stuff, right? So this takes in water, but it can also take in some random small solutes that we need from the extracellular environment. Receptor mediated endocytosis is basically like there's a very specific receptor, and that receptor is waiting for like the correct things to bind, and once the correct things bind, it will fold in and it will bring in a vesicle, and that vesicle is coded on the bottom. So what that means is that it knows where to go. Those little things are like signal molecules that take the metabolite to the right spot. Now, just because we can bring things in through endocytosis, we can also do the opposite. We can take things out through exocytosis, right? Very, correctly named. So in that sense, it's just a reverse process, right? The vesicle goes up to the plasma membrane, it merges with it, and then the cell can just dump whatever it needs to dump from the cell itself. So 2 ways of the cell interacting with its environment there. Okay, so now we have junctions. Junction. So junctions are a little bit hard to remember. So let me see. I think I have a good card. Junction. Yeah, maybe here. Maybe I should talk about cytoskeleton first, though. I should probably talk about cytoskeleton first, which is getting into. Actually, yeah, let's do that. Okay, I'm gonna put this off for later until after we talk about the cytoskeleton. Let's talk about the general idea of general biology, right? Like what is a cell, right? So we have Oh, okay. So for eukaryotic cells, right, versus prokaryotic cells, this is a big thing. Eukaryotic cells have a membrane-bound nucleus and membrane-bound organelles, and they have mitotic division, right? So cells, prokaryotes to eukaryotes, right? So basically, I'm sure you guys are familiar with most of the organelles, right? We have our nucleus, which is like the brain or whatever. It stores and protects the DNA. We have the mitochondria, which is the powerhouse of the cell, as it's known. And that's where we do energy, right? We learned about metabolism. We learned about how the energy is produced in the electron transport chain in the mitochondria. We know about the rough, and so there's two types of endoplasmic reticulum. There's rough and smooth endoplasmic reticulum. And so endoplasmic. So the endoplasmic reticulum, right, you can see it's very closely associated with the nucleus because here's the nucleus, right, and it has direct attachment to the rough endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it has ribosomes. It's studded with ribosomes, and proteins can be made here by those ribosomes. Whereas the smooth endoplasmic reticulum does not have those ribosomes. That's because carbohydrates and lipids are made there. So there's two different types of hormones, peptide hormones made from proteins and steroid hormones made from lipids. And this is where you get both of them, right? Oh, this is just showing like, the creation of cholesterol versus if you couldn't make cholesterol. But that's the point of the endoplasmic reticulum there. So rough endoplasmic reticulum, protein synthesis covered in ribosomes. Smooth endoplasmic reticulum, formation and breakdown of lipids, detox in the liver. In the liver, it handles detoxification, and it makes steroid hormones in the gonads. The ribosomes, as we talked about, right, help make protein. We talked about transcription and translation, right? How does that happen? Right? We showed that this is a ribosome in blue, right? And the ribosome just goes through these cycles of attaching, reading the mRNA and creating a polypeptide chain based on reading the mRNA, right? And we talked about this whole pathway of how this all happens, right? Once the mRNA is created, the mRNA comes outside, the ribosome reads it, and then it produces a protein based off of the template that it read. The Golgi apparatus. The Golgi apparatus, I always heard of this as like the UPS of the cell. So they modify and they package the proteins, especially for secretion. So what that means is like they can help with putting stuff into vesicles for movement out of the cell. Golgi. I have something about Golgi here. Oh, probably in protein transport. Yeah. So see, we made a protein, right? And so once the mRNA is converted into a protein, right? That protein can actually, depending on like so proteins can have little segments called signal sequences. And signal sequences will help get a protein to where it needs to go. So if a protein has a signal sequence of like, I actually need to be outside of the cell, I don't need to stay in the cell, right? Then it'll go to the endoplasmic reticulum. It will get converted into a vesicle. That vesicle will go to the Golgi, and the Golgi will, tag it with whatever appropriate signals it needs, and that will allow it to exit the cell completely and be secreted into the plasma membrane. It could even be just destined to be embedded in the plasma membrane rather than secreted like completely outside of the cell. It could, if a protein is supposed to go to the membrane, that's how it gets there, right? Because normally the ribosomes are just chilling, they're just around, right? So how does a protein actually go to where it needs to go? Through this process, through the Golgi. Lysosomes, like we talked about, are garbage disposal. So what they do is like they bind. So whenever a endosomes, yeah. So whenever we have this happening, right, whenever we have phagocytosis and something has come in, right, this vesicle will bind with the lysosome, and that it's going to destroy whatever is inside the, I can't find a good picture here. Lysosome. Yes, sort of. Basically, it's acidic on the inside, and it can bind to, it can kind of merge with the vesicles that come in to destroy whatever inside, or to degrade whatever is inside. And peroxisomes are kind of similar, and they metabolize lipids and toxins because they have peroxide in them. So, yeah. So these are some of the major organelles that you find in eukaryotic cells, right? We talked about the nucleus extensively when we talked about DNA, right? We talked about the nucleus has a double membrane, right? The nuclear envelope, and it's very regulated what gets in or out, right? The DNA is stored in chromatin, right? Wrapped around histones. We also talked about that, how DNA is stored, right? It's tightly wrapped, DNA packaging, right? It's tightly wrapped up inside the around these little histones inside the cell. It can be active or inactive, right? We talked about euchromatin is open and heterochromatin is closed. And we talked about why that happens when things are acetylated or closed versus open, right? It just depends on how tightly bound the negative DNA is to the histones. Now, mitochondria is also very, very important. Mitochondria is really probably the most unique organelle in there. Again, another double membrane, right? Inner and outer mitochondrial membrane. And the center, the very inside, is the matrix, and the in-between space is, intermembrane space, right? And so we talked more about that when we talked about the metabolism. Let's see if I can find. No, this isn't what I want. No, that's fine. Okay, so metabolism. Oh yeah, the other cool thing is that mitochondria has its own DNA. So there's sometimes like weird questions about that, like relating to reproduction and stuff. It's like, how was your mitochondrial DNA affected or whatever, right? And the trick there is that you get all of your mitochondria from your mom, because your mom provides the original, like the cell and everything, right? All you get from your dad is the sperm with his chromosomes, right? But all of the organelles come from your mom. So your mom provides, you only get the mitochondrial DNA from your mother, not from your father, because your father doesn't provide any mitochondria. So that's just a fun fact that they like to test on. The ER, right? The endoplasmic reticulum. We have the rough endoplasmic reticulum. Again, it's got ribosomes on it, so there's a lot of protein synthesis happening there. After proteins are synthesized by the ribosomes, they are passed through the rough endoplasmic reticulum to begin post-translational modifications and then sent off to the Golgi apparatus. That's what I showed you before. Where was it? Yeah, right here. Oh, this is what I was looking for with the signal peptide, right? So when there's a signaling molecule, that signaling molecule tells it to come here, and then it'll finish its production here in the rough ER. And the smooth endoplasmic reticulum, no ribosomes, and it just it helps with synthesis of lipids. Ribosomes are the site of protein synthesis. Pretty simple. We talked about eukaryotic ribosomes are different from prokaryotic ribosomes. Eukaryotic ribosomes have a 40 and a 60, and they're overall 80, I believe, and the other ones are 30, 70. Hold on. Yeah, a 40 and a 60, so they're overall 80. And the prokaryotic ones are 30, 70, and they're overall 90. No, that's not wrong. That's wrong. 30, 40, 60. Is it 30, 50, 70? I think it's 30, 50, 70. Yeah. I don't know why they want you to know that, but ribosomes. Anyways, yeah, that's the ribosomes are ribosomes. Yeah, 30, 50, 70, yeah, versus 40, 60, 80. Also, I was so confused, like why does like 40 plus 60 add up to 80, right? I was always like, I never understood it. And I was like, this makes no sense. But then I just randomly found out that the S is actually a unit called like this unit, right? And it measures density rather than weight. And that's why it doesn't add up properly. Again, another like random, like I was like, oh, okay. So that's why that is. I thought they were just like being weird for no reason. But I was like, oh, okay. So there's actually a reason. The more you know. Okay. So lysosomes we talk about, they have hydrolytic enzymes to break stuff down. Peroxisomes, they use peroxide to break stuff down. Cytoskeleton. Okay, so now we can explain everything that we were talking about before. So the cytoskeleton is very, very intricate, very, very a lot of things going on. The same way that we have a skeleton to hold up all of our tissue, right? The cytoskeleton exists to hold up the cells and provide structural support. But there's a lot of different components of the cytoskeleton. There's microfilaments, which are made of actin. There's microtubules, which are made of tubulin. Then there's intermediate filaments, which are just intermediate filaments. The roles that they have are different. Actin, right, microfilaments help with the movement of the cell, the endocytosis and exocytosis, cleavage, and muscle contraction. So these guys are the reason that this works this way. I'll show you here, microfilaments or cytoskeleton. Yeah, this is a good picture. So you can see that there's a variety of different things, a variety of different cytoskeletal fibers throughout the cell. And so the structure of them is also different. Okay, yeah. So microfilaments are more for the movement of the cell, the endocytosis, exocytosis, right? So when we see this happening, right? How is this actually happening? How is the membrane actually reaching up like this, right? How is that actually happening? Well, it's because there are the little fibers in there that are actually pushing the plasma membrane out like that. It's kind of like if your bones, if you think about like when you're moving, it's like you're not moving, but your bones are moving and your tissue is just following it. If you think about it that way, that's essentially what's happening there. And so so that's what microfilaments are doing. Then microtubules have maintained the structure. They also make up cilia and flagella, and they help with intracellular transport, right? So this is very important. I think I showed you guys this before, but we have these two types of transport that runs along microtubules. We have kinesin, which moves stuff away from the center of the cell, and dynein, which moves stuff towards the center of the cell. And so I think I showed you, I'm pretty sure I showed you guys the GIF of like the little kinesin walking on the microtubule, which is pretty kind of fun. And so then we have the intermediate filaments, and these guys are just for structural support, cellular adhesion, for example, keratin, like you have in your nails and stuff. And so now let's talk about, so right after this, we're going to talk about the junctions, but basically the cytoskeleton originates from a region close to the nucleus called the microtubule organizing center. And it also contains centrioles where things like the cilia form. So you can see here, the centrioles right here near the nucleus, and that's where it all stems out from. And I want to show you guys, yeah, so we have the junctions and we have, okay, good. So in a nutshell, this is like my huge summary of everything to do with this, right? So the cytoskeleton is a network of protein fibers that helps with cellular movement and maintaining the cell structure. The key differences: the microfilaments are composed of actin. They're also called actin filaments, right? The microtubulars are composed of tubulin, right? So that's a key difference. Then we have the intermediate filaments, which are just proteins like keratin. You can also consider that the microfilaments are micro, they're small tubes, whereas the tubulins are large hollow tubes, and the intermediate filaments are like kind of medium. In terms of roles, microfilaments help with muscle contraction and movement, right? Think about how they help with cell eating, phagocytosis. They help with when you contract your muscles. We'll explain more about that. Whereas microtubulins are not moving anything. They are just there as tracks. And so intracellular transport happens on top of the microtubules, but the microtubules themselves are not what's moving around. The microtubules, it's like a train running on a track. That's what a microtubule is. Whereas the microfilaments are actively actually moving around. Then we have, what else do I want to talk about? So then we can, okay, yeah, now we can talk about the tight junctions and the desmosomes that we were talking about before. So the tight junctions and the desmosomes, oh, I gotta close this. So the tight junctions and the desmosomes do different things. Okay, so intercellular junctions are regions of contact between the plasma membranes of two or more adjacent cells. So when cells are directly next to each other, this is a way for them to interact with each other directly. So first off, we have, so the ones that we have talked about are, first let's start with actin filaments. Tight junctions provide an impermeable barrier to separate environments by actin microfilaments. And we see this in epithelial barriers like the blood versus the lumen in the gut. Anywhere where you would need like a barrier between two different environments, you have tight junctions. So you can see that tight junctions kind of look like stitching, like the cell plasma membranes are being stitched together, like sewn together. And what that does is that it prevents stuff from going through this cell layer. So if there's like acidity, if this is like, just pretend that this is the stomach, and like if there's like acid up here, right? If there's acid up here, obviously the acid needs to be stuck in the stomach, and we can't have it spilling out into like the surrounding area, right? So the tight junctions are there to keep a tight seal, to seal those cells together, so that things can't, go where they're not supposed to be. Number one, tight junctions. And tight junctions are what? Are made from actin filaments. Number two, desmosomes. Desmosomes provide strength by binding adjacent cells through the intermediate filaments of their cytoskeleton. So, and you can find this in muscle tissue or epithelial skin. So what does that mean? The desmosomes are right here, right? And what they're doing is they're providing strength by basically linking the cells together. You see how they're kind of linking the cells together? So they're, in a way, they're kind of like making a net, like a mesh throughout all the cells. So basically you can't just punch a hole through one cell because it'll be linked to all the other cells. And it kind of provides more strength to the whole system, to the whole layer of the cells. So yeah, those are desmosomes. And desmosomes are made out of intermediate filaments. Now, lastly, we have gap junctions, which are different. Gap junctions just allow cell-to-cell communication through protein channels. So what we have here are gap junctions, and they basically, it's in the name. They provide a gap between cells, so things can move in. Like solutes can be passed along through cells, right? And this is kind of how this is part of how like an action potential can travel, because a signal in one cell can quickly get passed into another cell, and so on and so on. Those are what gap junctions are. But they are selective. They're not just like willy-nilly passing anything that they want, right? They are selective to certain things. So they can be open and closed. Okay. So that was overall. So overall, that's, let me see, where are we at? Flagella and cilia. Oh yeah, I can also talk about flagella and cilia. So we talked about kind of their role in the cell. They also have a role in no. I had a picture that was nice. Hold on. Flagella. Interesting. No, I don't seem to have that picture. Basically, there's a in eukaryotes, the flagella help. So the flagella is like a little tail, right? And like you see it in sperm, and inside that thing, right? They're powered by ATP and stuff, but there's a microtubule structure that has, yeah, the 9+2 structure. So let me find this. 9+2 structure. This, okay. So the microtubules are in this kind of arrangement. They have a double in the middle, and then they have 1, 2, 3, 4, 5, 6, 7, 8, 9 pairs on the outside. So one pair in the middle and 9 pairs on the outside. In my opinion, it really should be 9+1 because they counted pairs, but here they counted pairs and then here they counted them individually. Why did they do that? I don't know. Maybe I'll find out someday, but 9+2 arrangement. Okay, anywho, so yeah, those are basically all the organelles and things to understand about cells in terms of organelles. Any questions about that so far? Let me see. Oh, so I think we can dive a little bit deeper on that. Especially on mitochondria, we can dive in deeper. So in mitochondria, yeah. Let's see. This and then tissue cells. Yeah, okay. So I do want to talk a little bit more about the mitochondria because it does come up a lot. So major distinguishing feature from prokaryotes to eukaryotes, right? Because we have organelles and they don't. We have the mitochondria, and this is where the ATP is produced, right? Talked about that. And the matrix contains the mitochondrial DNA. Mitochondria are self-replicating. They're really quite weird. They're like little aliens inside of our own cells, right? They're just doing their own thing. And this is what it looks like. So the structure is important, right? The matrix is inside, and then the intermembrane space is in between, and then the outer membrane is outside, right? And these infoldings are called cristae. Why do we have these infoldings, right? This is a really good question. Why do we have these infoldings? We have to consider, remember, structure and function are always accomplishing the same goal. The structure is the function is determined by the structure. So we have these infoldings. So basically, first off, what are we actually doing in the mitochondria? So let's think about this. Let's work backwards. We know the function, so why is the structure that way? The function, right, as we talked about, why is it what's the meme about mitochondria, right? It's the powerhouse of the cell. So what do we do in the mitochondria? What's the point of mitochondria? We make ATP, right? From the where is it? Oh, not here. From the electron transport chain. So in this whole situation, we are pushing protons into the intermembrane space, and then they're coming back down, and that's allowing us to make ATP. So we're using the membrane, right, to make ATP. When we do that, so imagine if the inside of the mitochondria was just a circle, was just another circle, right? The reason that one is better than the other is because having these infoldings is giving us more surface area. When we have more surface area, that gives us more sites to actually do the electron transport chain. And if we have more area to actually do the electron transport chain, then we can make ATP more effectively. More ATP is going to be made because we just have more space to actually do it. So that's why we have these infoldings. So remember, always think about every structure has a function, and every function has a structure behind it. So always think about what is the role of the structure here, and how is it informing the function. Okay. The nucleus. Oh yeah, this is nice. So the nucleolus is where all the chromatin is like kind of balled up and condensed, right? We have the nuclear envelope, which is again another bilayer, right? That prevents stuff from getting in and out. And then we have this is a rough endoplasmic reticulum, right? Yeah, it is. This is a rough endoplasmic reticulum with the ribosomes around. That's where we have our DNA. Okay, so now cells. So now we've talked about the inner workings of cells. Now we can talk about kind of like the outer types of cells. I mean, the whole cells. So epithelial tissue is a very common type of tissue throughout our body. It's basically like surface cells. So, but the weird part is, like, as a human, right, you're kind of like you have two surfaces. You have an outer surface and an inner surface. So in a way, you can think of the person as like a very weird long donut. So for example, if I draw, let's say I draw, okay, I'm not going to draw like, okay, so let me just draw a person like this, right? Okay, this is really bad, but just imagine that this is a person. And I forgot to draw their arms, so whatever. There's their arms. And then this is, and then that's their mouth. So as a person, right, we have surface on the outside. However, when we eat stuff, right, and when we breathe stuff, we have, you know, another track running on the inside, right? So both of these, both like our skin, right, on the outside, our skin on the outside is exposed to the environment, but on the inside, right, we have a long tract that's also exposed to the environment, just in a different way, right? And that's different from like the in-between, right? So the in-between would be like right here, because this is not being exposed to the environment directly, right? So overall, the point is that we have epithelial tissue to to at these barriers, at these barrier areas that are interacting directly with the environment. So they are so that inner part, right, is like the lumen. The lumens are like the hollow areas. So epithelial tissue covers the outside of organs and structures in the body, and they line the lumens of organs in a single layer or multiple layers of cells. And epithelial tissues have a variety of different classifications. Squamous epithelial cells are round and flat, and they have a center nucleus. Cuboidal cells are obviously cube shaped, right? Usually in a single layer. Columnar is tall and kind of narrowish. Let me see. Yes. So here we have simple squamous epithelial tissue, right? Cuboidal epithelial tissue, columnar, long and tall, pseudostratified. So this is normal, simple columnar epithelial tissue. Pseudostratified is that it looks like it's all over the place, but actually you can see that they're all still columns. They're all still like single file columns. It's just that the nucleus is in different spots. Now this is stratified squamous epithelial instead of simple, right? We have stratified squamous epithelial tissue, stratified cuboidal epithelial tissue, stratified columnar epithelial tissue right here, and transitional epithelial tissue. So you can see that there's a transition because this is kind of like pseudostratified, but then this becomes cuboidal. So there's a transition between them. So these are just different. So now we're kind of getting more into the anatomy side of the MCAT, where you're kind of being able to identify certain anatomical traits and where you might find them, what value they have, what do they do, etc. Right? So, for example, columnar epithelial cells, right, in the digestive tract, absorb material from the lumen and prepare it for entry into the body, right? So that's basically something that you can, for anatomy, it's like the part of the MCAT that's most about memorizing and most about just drilling down like what everything actually is and how it works. Yeah. So then goblet cells, yeah, that's just another type of cell that you see where they secrete mucus. I actually don't know why it's called a goblet cell, but they secrete mucus. So you could see that like, throughout that digestive tract basically. And yeah. and transitional cells. Oh, only in the urinary system. Okay. So connective tissue is another type. It's not, so it's a bunch of fibers, basically. It's basically a bunch of fibers. Connective tissue. Yes. So we just talked about epithelial tissue, right? Epithelial cells, right? Then there's connective tissue. And in a sense, bones are connective tissue. Bones are actually considered connective tissue. Bones, blood, fat, tendons and ligaments, and cartilage are all types of connective tissue. And basically, their goal is to like connect tissues, right? To connect things. In terms of bones, like tissue is attached to our bones and like stuck on them, right? In terms of tendons and ligaments, they link muscles to bones, or they link bone to bone or bone to muscle, right? So these are just linking different things that link our bodies together. And most of the connective tissue is just extracellular matrix. So extracellular matrix is basically saying it's collagen, elastin, all these different kinds of proteins that are, they're just kind of filler. It's kind of weird to visualize, but there's just a bunch of stuff in our body that's there to connect everything else. And yeah, I don't know if you need to know this specifically, but the principal cell of connective tissue is a fibroblast. Yeah, you do need to know that. So these guys are, the cell makes the fibers in the first place. So fibroblasts, right? And yeah, it just looks like it's kind of just a mess of different fibers and whatnot. And you can see here, like the fibroblasts are producing these fibers. Oh, this goes into cartilage and stuff. Yeah, actually, there's two different types of cartilage. Yeah, this is kind of interesting. Cartilage is made by chondrocytes, right? And cartilage with few collagen. So there's two different types of cartilage: hyaline cartilage and elastic cartilage. So elastic cartilage is a type that you have in your ears or in your nose because you can wiggle it around, right? Whereas there's fibrocartilage, which you don't wiggle around. That is the cartilage that you find between your bones, like covering your joints and stuff to prevent like your bones from rubbing together. So yeah, you can have, wait, hold on. Cartilage can also transform when a hyaline cartilage is found in movable joints such as the knee and shoulder, or becomes damaged. The damaged hyaline cartilage is replaced by fibrocartilage. Wait, oh, okay, well, hold on. Fibrocartilage, the stiff cartilage, intervertebral discs. Sorry. Okay, so discs, you're not moving them, right? So that's where where you get the hard the hard cartilage, which is the fibrocartilage. The elastic cartilage, and the hyaline cartilage, where is it? Hyaline cartilage, right, is what you would find to reduce friction. Yes. So hyaline cartilage is found at the ends of the long bones, which reduces friction, right? That's hyaline cartilage. Then you have elastic cartilage, which is in your ears and your nose. Then you have fibrocartilage, which is hard cartilage that you find within your bones. That's the different types of cartilage, essentially. Okay. So now I think we've pretty much covered 2A. Yeah. So now we're gonna get into a practice passage, which kind of touches on some of the stuff and some not of the stuff. Where did I put it? Oh, we also have prokaryotes versus eukaryotes. Yeah, we have a little bit of time. Okay, let's do that then. I talked about this. Okay, so prokaryotes and eukaryotes, right? It is definitely important to distinguish between the two types of, and even though you would think like, like, oh yeah, this is prokaryote, this is eukaryote, right? It can kind of be tricky sometimes because they'll maybe only give you a very narrow slice of all the information that you could have. So the main things that we talked about are eukaryotes reproduce using mitosis and meiosis, whereas prokaryotes reproduce with binary fission, right? Let me see if I can find, maybe I have a card about binary fission. No, I don't. Oh, yeah. So cells, I mean not cells, prokaryotic cells, they have binary fission, which means production of identical daughter cells like this, right? They just duplicate their chromosome, then they elongate and they split. It's very simple. It's not complicated like mitosis and meiosis is. And we're going to talk in depth about mitosis and meiosis. Major difference in terms of organelles. No membrane-bound organelles. They still have ribosomes because ribosomes are mostly just free-floating, but no membrane-bound organelles like lysosomes, endoplasmic reticulum, they don't have any of that, right? They don't even have a nucleus. They just have a nucleoid region where they keep their DNA. Whereas eukaryotes are very specific and they have, specific functions with organelles. Circular DNA versus chromosomal DNA. We talked about the different ribosome densities. They transfer their DNA through a couple of different steps called like transformation, transduction, and conjugation. So let me see if we can talk about that. Transformation. Yes. So the ways that cells can, I mean, not cells, the ways that bacteria can get genetic diversity, right? We saw that they just do binary fission. So they just duplicate their chromosomes and then they make new ones, right? So how can they get genetic diversity? How can they, for example, how do some, like, let's say you have a petri dish, right? And you introduce some antibiotics, right? Most of the cells will die. Most of the bacteria cells will die, right? But some of them could survive if only they had like a certain antibiotic trait, right? So how could they possibly get that? Well, there's a couple of different ways. One option is called transformation, and that's basically where another bacteria died and its DNA is just floating around, and a bacteria might just pick it up. It might just say, oh, this is interesting, and just pick it up, and it could get integrated into its DNA. And so now you'll see here again the difference between structure and function, right? Or not this difference, but the theme of structure and function. If the bacterial DNA is just chilling out here, no membrane covering it, nothing hiding it, right? Then it's very easy to access, and it's very easy for it to change it up, right? So that's a big difference between prokaryotes and eukaryotes. Eukaryotes want to maintain their DNA. They really don't like to have it changed too much and changed like without their control. Whereas prokaryotes are kind of open to it. They will accept various different ways of being altered or changed. Another option is transduction. So transduction is when a bacteriophage, right, takes it does its viral replication and it lyses the cell. when it lyses a cell, it can accidentally pick up some of the bacterial DNA instead of picking up viral DNA. So essentially, the bacteriophage comes in, it injects its DNA, and then it forces the cell to reproduce its own viral DNA, right? But then when it reassembles, some of them might accidentally pick up the host DNA of the bacteria. So now they're actually now, instead of injecting viral DNA into another cell, they've actually just injected bacterial DNA into another bacteria. So now this prokaryote has some new prokaryotic DNA. And who knows, maybe that was the gene for antibiotic resistance. So maybe now this bacteria is defensive against antibiotics. That's just another avenue that it could gain some genes. Conjugation is the last one. Conjugation is interesting because it's kind of like it's kind of how we would see reproduction for it's sexual reproduction for bacteria, basically. So it's the direct transfer of genes from one cell to another cell, and it will increase diversity, but it doesn't actually reproduce anything. So this guy comes in and he has an F factor. F factor means that he is able it is able to reproduce and pass on its chromosomes. So it has a sex pilus, which it attaches to another cell, and then it can transfer the F factor and also sometimes like some plasmids, right? By doing that, by passing it on, then this one is now also able to conjugate with other bacteria. So it kind of passes on like a not really like a virus, but it passes on. So these are 3 different ways that bacteria can gain genetic diversity, in contrast to how we gain genetic diversity through meiosis and through crossing over and all this kind of stuff with our chromosomes. They'll just take whatever. They'll just take whatever they get from anywhere. Yeah. And so specifically the F factor is a circular DNA element that allows them to perform conjugation. If they don't have the F factor, then they're not they can receive conjugation, but they can't construct the sex pilus to actually perform conjugation on another cell. Yeah, so that's bacterial. That's what they meant by transfer of DNA through transformation, transduction, and conjugation. 3 different paths. For us, we do it through sexual reproduction, aka meiosis. Prokaryotes have a cell wall made of peptidoglycan. We don't have a cell wall in our animal cells. We don't have a cell wall, but they do have a cell wall. And flagella structure. Okay, so this is kind of a niche difference, but there is a difference in the type of flagella that we have. So, and again, we talked about the cell wall. This is the peptidoglycan, and there's also 2 different kinds of cell. There's gram-positive cells and gram-negative cells. What that refers to is a process called gram staining, where when you apply certain chemicals, again, this is like the lab side, which we'll talk about, but basically when you apply certain chemicals, some bacteria will stain purple and some will stain pink. And it depends on what structure they have. If they have a double membrane like this, or if they have a single membrane and a thick peptidoglycan wall. So, and the gram-positive ones, which have the thick peptidoglycan cell wall, stains purple, and the gram-negative ones have a thin cell wall, but they have 2 membranes, and these guys stain pink. And the difference is that the gram-negative bacteria are the ones that are able to participate in conjugation. Flagella. So the main point here was that there's kind of 3 different parts of the flagella. There's a basal body, which is like the motor, which causes the spinning motion. There's a hook, which kind of curves it a little bit. And then the filament itself, which is the long tail, which allows the bacteria to move around. So, yeah. Oh, and then we talked about the different types of cells. We talked about gap junction and that. Stem cells. Oh, yeah. So stem cells. So this will get more into reproduction. With stem cells. Oh man, immune system is gonna be one heck of a topic. Immune system is quite in depth. Okay, I don't see stem cells, but basically, stem cells are unique to eukaryotes, right? And they're cells that are undifferentiated. So they can, so the reason that you hear so much about stem cells in like the news and whatever is because stem cells are cells that have unlimited potential. They can be whatever we want them to be if we figure out how to make them grow properly, right? But basically, when you start, you have a bunch of stem cells, and those stem cells will eventually differentiate into various organs and organ systems. So you may have seen like cell lineage lines, stem cell lineage. Yeah, stuff like this. So you see, you start with an embryonic stem cell, and then they split up into three different major ectoderm, mesoderm, endoderm, and those will keep differentiating until they become a very specific type of cell. But they all started from the same kind of cell, the stem cell. And so stem cells, um, at the beginning, right, they're called totipotent because they can become any kind of germ layer. So the germ layers are here: ectoderm, mesoderm, and endoderm. This is kind of, I guess we can just talk about this now. Endoderm. So in the cell, actually, this is more towards reproduction, but basically, there's three different types of. So this is like the original what your cells look like, and these are so right now these are all not hold on. They're not oh, okay. So basically, there's three different germ cell layers, right? Originally, they're all totipotent. Originally, they can all be anything. They can originally all be anything. Where is it? They're originally all totipotent, total potential, right? They can become any kind of germ layer. Once they become a germ layer, once they commit to becoming ectoderm or mesoderm or endoderm, they're now considered pluripotent. So they're pluripotent because they can become any of those, any of the systems downstream of this, but they can't go backwards, right? Or they cannot, a mesodermal cell cannot suddenly produce something from the ectoderm, right? They have committed in one way. And then you get to multipotent stem cells, right? When they have committed even further, and they are still stem cells because they can make a variety of different cells, but only the category of cells that they already specialize into. So for example, platelets or red blood cells and white blood cells, those are all under the umbrella of hematopoietic stem cells. So you can see as you specialize further, your options are more limited, like in terms of you are getting narrower and narrower, but you can differentiate into different things. So everything starts totipotent, becomes pluripotent, becomes multipotent, and then finally becomes, the whatever cell it needed to be. But yeah, stem cells are very, very under heavy research because if we can figure out, how to effectively take totipotent stem cells and grow them into, say a liver, grow them into a kidney, right? If we can do that, then that solves a lot of problems that we have, right? We could even, like, for example, we have stuff like stents to expand arteries and but what if we could just grow another artery and replace it, right? Like that's kind of like the realm of science fiction, but we hope to be able to do that one day. So overall, so that I think that's overall what we wanted to talk about for this section. And now we'll get into a passage. Now we'll do some passage. And this is, so I was looking at this passage. I think it's a nice passage because it's a little bit complex and a little bit dense. So here's what I want to do. Let's take this, and it does have some questions about the cell stuff that we talked about. So I'm gonna post, what time is it right now? It's 11:47. Okay, so I'm gonna post this, and again, I want everybody to like, uh, tackle it and give it a shot because that's how you're going to learn. And we'll give it, let's give it 11:50, 12, 1, 2. Let's give it until like 12:20-ish, I think. I think take some time, or maybe we won't need that much time. Let's aim for 11:15. So I want everybody to open up that link and go to biology passage number 1 and give it your best shot. And if you have any questions while you're reading, you can ask me. But overall, let's try to go through it, and then at 11:15 we'll go through it together, and hopefully we'll answer some questions, and this will be good practice for a bio passage. Anybody have any questions? Okay, so then we're going to, okay. So in this passage, we have a very classic kind of passage that starts with a little bit of background of some kind of, not metabolic, but biological disease or condition, and then some research that they did about it. And then we have to answer some questions about it, right? The interesting thing is, so let's read this passage first, right? I'm just going to read it first. I'm not going to try to interpret anything. We're just going to read it first. Postoperative acute kidney injury, AKI, is a severe complication after liver transplantation, as its deterioration and magnification can lead to an increase in mortality. Connexin 43 mediates direct transmission of intracellular signals between neighboring cells, always considered to be the potent biological basis of organ damage, deterioration, and magnification. Researchers obtained donor liver tissues of patients in order to explore the effects of CX43 on organ damage. Reperfusion is a biological process used to refill blood in AKI. Donor livers displayed more severe lobular distortion with necrosis, apparent edema, hemorrhage, and neutrophil infiltration after reperfusion, compared with donor liver tissues obtained before reperfusion or normal liver tissue from patients with hepatic hemangioma. After reperfusion, CX43 mRNA and protein levels were determined as shown in Figure 1. Autologous orthotopic liver transplantation, AOLT, rats were built by researchers in order to further study AKI. Researchers hypothesized that the change in CX43 expression might play an important role in AKI following AOLT. Researchers used heptanol, a well-known inhibitor of CX43, without hepatotoxicity to alter the function of junctions composed of CX43. Heptanol had no effects on CX43 expression in kidneys or livers. Heptanol or a sham control were injected intravenously into the rats, and the pathological score of kidney cells was determined to analyze disease progression. The results are shown in Figure 2. Researchers also believed that the RIP1 may be involved in CX43 function, and they confirmed the interaction of the proteins through co-immunoprecipitation. RIP1 is a typical marker of necroptosis. So we read all of that, and it was pretty in-depth and confusing, right? So what is the very first question that we get about this? What type of junction does CX43 most likely form? So what we have here is a lot of like heavy experimental questions, right? Heavy experiment and or not questions, heavy a passage that's heavily based in experimentation. But what's the very first question? The first question is about what type of junction does this come from? So what is this asking about? This is asking about anatomy, right? This isn't asking you to do any kind of experimental analysis or comprehension. In this case, we're just trying to understand some anatomy. So if that's the case, right? Well, what do? Rather than look at the graphs, right? Let's try to understand what is CX43, right? And so they say connexin 43 mediates direct transmission of intracellular signals between neighboring cells. So then which of these options sounds the most like that? Gap junctions, tight junctions, desmosomes, hemidesmosomes. So we talked about when we looked at desmosome, when we looked at the various types of, where's the good picture? Yeah, so we talked about the various types of cell junctions, right? And the tight junctions are there to make a tight seal, right? The desmosomes are there to anchor everything together. And the gap junctions are there to allow molecules to pass through, right? So which of these sounds the most like CX43? Mediates direct transmission of intracellular signals between neighboring cells. So out of these three, what do we think is the most like to be most like direct transmission of intracellular signals? Is that a tight junction? Is that a gap junction? Or is that a desmosome? Right, it's a gap junction. The gap junction is the one that allows molecules to pass between neighboring cells, right? So notice that even though this passage is quite in depth, right? The first question is merely testing your content knowledge. This is a discrete that is embedded in a passage. And this is pretty common. Like it's not, most passages that you get are not hardcore experimental analysis. They're mostly questions like these, and then a little bit of experimental analysis. So we could spend time like trying to understand the passage completely, but rather it's better to kind of get acclimated, just read the passage through, then look at the questions to see how much they're demanding from you. How much do they really want you to understand the passage? Or rather, how much can you get away with not knowing the passage? For example, I mean, like you have like 10 or 11 different passages to break down, or is it 10? Or is it 9 or 10? One of the two. You have some number of passages to break down, right? And you could, write a whole paper about each single passage, right? But if you don't need to, then you don't have to, right? So only dedicate as much time as you need to in order to answer a question. So in this case, the answer was right here in the second line. We didn't have to pour over graphs. We didn't have to do anything like that. So, oh, also another good habit, right? Hemidesmosome. I don't think I covered this. I remember that this is something about basement membrane, but I forgot what it is. Hemidesmosomes are similar, but their main function is to attach epithelial cells to underlying structures. So you see, we learned three things, right? We knew what tight junctions were, we knew what gap junctions were, and we knew what desmosomes were. But we had not heard of hemidesmosomes. Remember, this is when you're doing problems, this is a learning experience for you. If you see something you don't recognize, Google it, look at it, try to figure out what it is. And then if you feel like it's important enough to memorize, just put it in your Anki cards or put it in your notes or whatever, so that you picked it up. Okay. So this is a gap junction because they said direct transmission of intracellular signals between neighboring cells. Then A researcher discovers a new CX43 inhibitor made of a glycerol backbone and 3 saturated fatty acid tails. Which of the following describes the path by which the inhibitor enters the cells? So again, this is not we still are not talking about the experiment. We're now just talking about this random hypothetical of a new inhibitor and how can it enter a cell, right? So glycerol backbone and 3 saturated fatty acid tails. Does anybody know what that is? What is that molecule? We talked about it a lot when we talked about metabolism. Glycerol backbone and 3 saturated fatty acid tails. It's not cholesterol. So cholesterol is a different kind of lipid. The glycerol backbone and 3 saturated fatty acid tails is a triglycerol. Triglycerol. So the 3 fatty acid tails. And so we talked about this in metabolism because we have these long fatty acid tails. We got 3 of them. And then we have the glycerol backbone to which they're attached. Now, cholesterol is quite different in terms of structure, even though it's a lipid, even though they're both lipids. Lipids are kind of a big group of things that are nonpolar. That's it. But in terms of the structure, they can be very, very different. So the cholesterol structure is actually a ring structure rather than a chain structure. So cholesterol has these 4 rings, and cholesterol is not again, we talk about structure and function, right? Structure and function, structure and function. The structure of triglycerols is set up this way because when we metabolize it, we break down these carbons bit by bit. So we break them down in 2 carbons at a time. Break, 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 break. And every time we break it, we're getting energy. So that's why the structure of this is good for energy breakdown, which is what it does. Whereas cholesterol is ring structure. And those ring structures, this structure is good for building hormones. So we have different hormones that are cholesterol based in terms of structure. So yes, so what was I talking about? So yes, glycerol backbone and 3 saturated fatty acid tails, right? So again, let's think about structure and function, right? We have a glycerol backbone and 3 saturated fatty acid tails. This thing is it polar or non-polar? Where are we looking at? Triglycerides. Here. Here. So actually, I don't like this picture. I want a uniform one. This one's okay. So we have a glycerol, right? Where can I draw? This molecule here. So here on the glycerol, right? We have some O's, some OH groups, right? And on the fatty acid, we also have some OH groups, right? These fatty acids. And when they combine, they look like this, right? However, there's two different regions, right? There's a glycerol backbone here, and then there's these fatty acid chains, right? So the chains are different from the glycerol. The chains are just straight up long hydrocarbon chains. And those chains are just carbons, right? So when you have these long carbon chains, these guys are nonpolar. Now, you can the glycerol, right? You can say it has a little bit of polarity in the inside where the oxygens are. But overall, if we're considering overall, right? These long carbon chains are so nonpolar that like it kind of overrides any slight polarity that you might get from the glycerol. So we're just thinking about overall how a molecule, where a molecule wants to be, how it wants to exist. This thing has long hydrocarbon chains or rings, anything hydrocarbons, anything with these just carbon to carbon to carbon to carbon to carbon, we're going to consider as nonpolar overall. So when we have something like this, so basically we're starting with our fact of we have a molecule and we consider that it's nonpolar because of the fatty acid chains. Now the question that they're asking, right, is which of the following describes the path by which the inhibitor enters the cells? So now they're basically asking, we have this nonpolar thing, how would it enter a cell? So we talked about the different types of channels and stuff that exist for molecules to enter, right? Is the bilayer, is the plasma membrane polar or nonpolar? The plasma membrane. Or I guess that might be a little bit of a tricky question if you consider what part of the plasma membrane you're looking at. Here, right, obviously the top part of it, the outer side of it could be considered polar. But when I say the plasma membrane, I'm talking about the inner part that we have to cross through. And the inner part is nonpolar, right? It's hydrophobic. So if something is nonpolar, right, if a molecule is nonpolar, and then the plasma membrane itself is also nonpolar, then that means that it's okay passing through it. It would be okay passing through it. So a nonpolar molecule can pass through. Oh yeah, wait. I had a picture that explained this exactly. No, not this one. Oh, was it active transport? Yeah, I think it was. Here, we said that non-polar solutes can transport directly across the membrane because of that non-polar, because of the non-polar bilayer here, right? So if the molecule was polar, it would need a carrier protein. For polar stuff, they need to go through a carrier protein. For non-polar stuff, they can just go straight through. So if we're looking at this question, they said a glycerol backbone and 3 saturated fatty acid tails. So that means that that thing overall is nonpolar. So if it's overall nonpolar, would it go through a receptor or would it go straight through, right? And again, we can also look at, if we just look at the anatomy of this question, right? This says a receptor, right? This says a receptor, right? Some kind of protein, right? And this says a channel, which is a protein, right? This is the only one where they're not talking about a protein. No protein. So again, so this is like the odd one out, right? That is just another way to kind of see the pattern. But overall, because the glycerol backbone and 3 saturated fatty acid tails is a non-polar molecule, it can diffuse directly through the cell membrane. And that's exactly what we're seeing in this image. A non-polar molecule can diffuse directly through the membrane. If it was polar, it would have to be one of the other choices. A polar molecule would have to use some kind of protein or receptor to get through. Okay. So that was two questions, and both of the question, neither question actually asked us about anything to do with the experiment. It was simply about some anatomy knowledge and some understanding of osmosis and diffusion. So now we have, let's see, researchers decide to interrupt this interaction in the AOL2 rat model without using a small molecule inhibitor, which of the following will most likely disrupt the interaction. And so what did they say about this interaction? They said, researchers also believe that the RIP1 may be involved. How did I know that the inhibitor was hydrophobic? So the reason that I know the inhibitor is hydrophobic is because, so let's talk a little bit about hydrophobicity. This is in terms of amino acids, but I want to explain more in terms of, yes, because it's a triglyceride. But I also want to explain it from like, just in a general sense, right? So because they said 3 fatty acids and glycerol, right? If you know that 3 fatty acids and glycerol is a triglyceride, and you know that a triglyceride is hydrophobic, then those are facts that can tell you, right? But what if you don't know that? Well, starting from a basic fact of what hydrophobic versus hydrophilic actually means, right? Or what nonpolar versus polar actually means. Nonpolar versus polar bonds. This is kind of, I mean, yeah, this is, but also I don't want to explain it in that way just yet. I'm trying to explain. So there are some molecules, there are some atoms that are electronegative, right? And so, yeah, okay, this is fine. Oh God, this is a horrible image. I don't know why it looks like that. So anyways, in this case, there are oxygens here, right? But because they're both oxygen, they both have equal pull on the electrons, and therefore this is nonpolar. Even though it's oxygen, and oxygen is electronegative, you have to consider the structure of things. This is nonpolar because they both have an equal pull on the electrons. Whereas in water, the oxygen is much more electronegative than the hydrogens, right? And if you're curious, like what is electronegativity, right? We'll talk about that when we get to general chemistry and electronegativity trend. When we talk about electronegativity, if we look at the periodic table, electronegativity increases going towards the right of the table. So the most electronegative stuff is like oxygen and fluorine. So if we're just looking at, between oxygen and hydrogen, oxygen would be the more electronegative one. And then we have this little symbol, the delta, and then a negative, the charge, right? So delta means partial. So this guy is not fully negatively charged, but it's partially negative charged because the oxygen is able to take more than its fair share of the electrons away from the hydrogen because the hydrogen is less electronegative, less able to pull electrons toward it. So these guys are polar bonds. Now, the reason that the triglyceride is nonpolar is because the reason that a triglyceride is nonpolar is because in this carbon-hydrogen situation, first of all, carbon is not very electronegative, and this is all completely equal share. There's no reason for this carbon to pull more electrons than another carbon. And the hydrogens and the carbons are pretty share Hydrogens, when connected to carbons, are not really, you wouldn't really consider polar. Oxygen compared to hydrogen, yes, that's polar. But overall, this is so you can see the difference here, like within a phospholipid itself, because a phospholipid, which is what's being shown here, has a hydrophilic region and has a hydrophobic region. The hydrophobic regions are uniform, right? Uniform. There's no uneven electron sharing. The hydrophilic region has uneven electron sharing. It has a charged oxygen, right? This oxygen is taking more electrons than these oxygens are all taking electrons from that phosphate group. And this one is taking so much more electrons that it's fully negatively charged. So you can look at charge, but charge is not the end all be all. It's more like how uniform everything is. But I will tell you just in general, these long carbon hydrogen chains are hydrophobic and nonpolar. So that's just that's something that is a fact to know. Okay, good question. Thank you. So so phospholipid overall charged, so it's polar. Okay, good question. There is no such thing as considering a phospholipid overall because it has two distinct regions. We can't really say that a phospholipid is overall polar because there's a huge part of it that's non-polar. So that's why when we look at a membrane, phospholipid bilayer. When we look at a phospholipid bilayer, right, we cannot say overall that this is polar or non-polar. We can't because it has two distinct regions. It has a distinct region of polarity, polar head groups, in fact, it has two of those. And then it has a distinct region of non-polar, right? Non-polar tails here. So this is not polar, it's not non-polar, it's amphipathic. Amphipathic means that it has regions of both. Regions of both hydrophobic and both hydrophilic. Amphipathic. Amphipathic compounds. Compounds that contain both polar or charged and non-polar regions. The polar region dissolves in the water, but the non-polar region avoids the water. Make sense? Okay, good. So yeah, so these are all very good questions because these are like the basics, right? And if you start from the basics, it will let you understand new things that you run into. So clarifying the basics and having a good grasp of the basics is always a good thing that to aim for. Good. So now next time when you see like uniform things, you'll be able to think about, oh, okay, hydrophobic, hydrophilic. Okay, so where were we? So we were on this third question, I believe, and the third question is asking, researchers decide to interrupt the CX43 RIP1 interaction in the AOLT rat model without using a small molecule inhibitor. Which of the following will most likely disrupt the interaction? So what did they say about that? They said researchers also believe that the RIP1 may be involved in CX43. They confirmed the interaction of the proteins through co-immunoprecipitation. RIP1 is a typical marker of necroptosis. So one thing that we got here is that they are proteins, right? Confirm the interaction of the proteins. So these are both proteins, right? So that's going to make an impact on this question. Decide to interrupt the protein interaction in the rat model without using a small molecule inhibitor. Which of the following will most likely disrupt the interaction? So they're saying that they want to disrupt a protein interaction without using a small molecule inhibitor. So when they say small molecule inhibitor, so if you look at protein interactions, inhibition. I mean, if we just look at in general, I hope there's a general picture. Yeah. So if we look at in general, right, like if there are two proteins binding together and an inhibitor binds, maybe it will prevent like some kind of binding, right? Mm, this is a little bit more complicated than what I want, but basically, yeah, something like this, like the inhibitor binds, and now the substrate can't bind too well. In this case, it's just two proteins, like they are each other's substrate, but overall, they're saying, how can we do this inhibition without using an actual small molecule, right? So the four options that we have are siRNA for RIP1, radiation against heptanol, acetyltransferase specific for beta-actin, plasmid producing CX43. Now, plasmid producing CX43 doesn't make any sense because they want to interrupt this interaction. If we introduce a plasmid to produce CX43, isn't that going to increase the interaction? Because we'll have more CX43. We don't want more CX43. We want something that will inhibit this somehow. And making more CX43 is not going to inhibit this reaction. It's going to allow for more of this reaction. So this is not good. Acetyltransferase, an enzyme specific for beta-actin. I'm not sure what this has to do with anything. I don't remember when they talked about beta-actin, if they did. Acetyltransferase for beta-actin. Acetyltransferase. I'm not sure what that's about. But again, they're saying without using a small molecule inhibitor. I guess that's an enzyme, so I guess that's fine, but I don't really see what is the role of beta-actin here. They didn't say anything about CX43 or RIP1 having any involvement with beta-actin, at least as far as I can see. So that just seems kind of irrelevant to me. Radiation against heptanol. So what was the role of heptanol here? So researchers use heptanol, a well-known inhibitor of CX43. Now again, this also again doesn't make sense because we are trying to interrupt this interaction of CX43, right? Heptanol is an inhibitor against CX43. So if we destroy the inhibitor with radiation, then that's not interrupting, it's activating this interaction, right? Let me just write out my logic here, and you guys tell me if it makes sense. There's a CX43 interaction, right? And obviously anything that increases, so what will increase this interaction? What's going to increase this interaction? Well, it's going to increase if the amount of CX43 increases, or if the amount of RIP1 increases. It will lead to more interaction, right? So then they gave us that information that heptanol, right? Heptanol is a inhibitor of CX43. So if it's an inhibitor, that means when heptanol increases, then CX43 decreases. And if CX43 decreases, then the interaction between it and RIP1 should also decrease because there's less CX43. But this answer choice is saying using radiation against heptanol. So radiation is destructive, right? So if you use radiation to decrease heptanol, then it'll have the opposite effect. It'll decrease heptanol, which would increase CX43, which would increase the interaction. Again, this is not what we want. We want to interrupt the interaction. So this, again, does not make sense. Lastly, we have siRNA for RIP1. So now if you don't know what siRNA is, right, this is again another learning moment that you have to be like, okay, well, what is siRNA? And siRNA actually stands for silencing RNA. It is a small interfering RNA. And the reason that works here, RNA. Oh, no. Maybe I have it in here. Interfering. Yes, okay. So small siRNA is small interfering RNA and is synthetic RNA designed to degrade the complementary mRNA. So it is small interfering RNA. And so the reason that this works here is because they gave us the clue. The clue was the interaction of the proteins. How are proteins formed? Through transcription and translation, right? So we can use that small interfering RNA to go in and prevent the synthesis of the protein entirely. We can just prevent the synthesis. So for example, right here, right? Let's say that, the mRNA comes out and the mRNA is going to get attached to a ribosome and then get translated, right? What the siRNA will do is before the ribosome can come, the siRNA will come and it will bind to the mRNA. When it binds to the mRNA, it can degrade it and prevent it from getting turned into the protein. So if that happens, then there is no protein, right? And if there is no protein, if there is no RIP1 protein, then we'll decrease that will prevent the interaction. So, where's my, oh, I lost the text box, oh well. But anyways, did that logic make sense? Do you want me to go over the logic again? Does it make sense? siRNA? Okay. So now again, notice the main thing, all three of these still have not actually really asked us to investigate the experiment, right? Now we get which conclusion about CX43 regulation is best supported by figure 1. When you see something about this, which conclusion supported by figure, this is experimental analysis. Now they're saying, hey, look at the graph that we have and tell us, what do you understand from this? So now, notice how we can answer these questions with background knowledge, right? And we only need to focus and interpret the graphs that they ask us to, right? In this case, they're saying supported by figure 1, right? And that means that we don't actually need to understand figure 2 very well. So, be wise with your time. Let the questions guide you to what you should focus on, right? Which conclusion about this regulation is best supported by figure 1? Okay, so before I even read the questions, I know that I need to go back and I actually need to interpret figure 1 and understand what's going on here. Okay, so they said AKI, severe complication after liver transplantation, can lead to an increased mortality. CX43 mediates direct transmission, considered to be the potent biological basis of organ damage. So one thing very helpful to look at, right, is the axes, right? What are we measuring? Okay, we're measuring CX43 mRNA units normalized to normal liver tissue. Okay, make that simple. They're simply measuring CX43, right? Levels of CX43, right? In what? In normal liver tissue, in tissue before reperfusion in donor liver, and tissue after reperfusion in donor liver. Okay, so why are they doing that, right? Let's understand. Researchers obtained donor liver tissues, okay, I see the donor liver, tissues of patients in order to explore the effects of CX43 on organ damage. Okay, so they have just given us a lot of good information. We are trying to figure out the effect of CX43 on organ damage. It's now our job to figure out what the relationship is from the graph, right? Actually, the rest of this is kind of like not super important. Reperfusion is a biological process used to review blood and EKI. Donor livers displayed more severe lobar distension compared with donor livers obtained before. After reperfusion, CX mRNA protein level were determined as shown below. None of the rest of this paragraph gives me any information to understand this. None of it gives me any info about CX43 versus organ damage. Ultimately, it's the graph that gives me that info because I need to interpret the graph. That's what the test maker wants me to do. So they told me researchers obtained donor liver tissues of patients in order to explore the effects of CX43 on organ damage. Boom. When you see something like that, you need to write it down so that you know what it is that you are actually interpreting for, right? And so now here we can further break down like we can break down this graph, right? What we see is there are obviously different levels of CX43, right? And how does that relate to what happened? So in normal liver tissue, CX43 is kind of low. It looks like it's at 1. In donor liver, before reperfusion, it's at 2. And we can see that the little star here, or yeah, the star here tells us it's significant statistically, right? So it's increased compared to normal liver tissue in donor liver. And then after perfusion, it's even more increased, the amount of CX43. So that tells us that, you know, the donor tissue has increased CX43. And also that CX43 increases after perfusion, right? So this is basically the main stuff that you could get from this graph, right? And then if we go down, oh wait, hold on. There's also, oh yeah, there's also this little part. So this little part is kind of a how do I explain this? This will make more sense when we start talking about lab techniques, but this is kind of like a protein stain, or I think this is a Western blot. So it basically tells you what things what genes or proteins are kind of active, right? So they're showing us that in beta actin, these subunits are there, but in CX43, it's only for this one. So that's a little bit hard to explain. And it will only make more sense when we actually talk about how the test works and specifically. So, but we'll talk about that. Overall, you're asking which conclusion about CX43 regulation is best supported by figure 1, right? And we have this stuff about expression control occurring at transcription level or translational level. Donor livers have higher levels of CX43 after reperfusion when compared to normal liver tissue. That's actually what we saw, right? Because the CX43 increased after perfusion, after reperfusion, right? And that's exactly what we saw. The donor liver has increased CX43 compared to the normal liver tissue. So and then the other question is, normal livers treated with heptanol have lower levels of CX43 compared to donor livers. Now the thing is, this is only asking about figure 1. Heptanol only came up in figure 2. So it's already wrong to be considering heptanol. Let's focus on the figure that they actually asked us to focus on. So the answer here should be C. Now, in regards to A and B, I think what they're getting at is like using this to explain like transcription versus translation versus the mRNA. But I don't think you can really draw a conclusion from here because there's not really a a clear pattern like there is here with the CX43 increasing with the perfusion. So C is the best answer there. So we can just take a look at our answer choices. So we had said that it's a gap junction, right? This is anatomical knowledge. We had said that it diffuses through the cell membrane. This is a little bit of knowledge about, what triglycerides are like, a little bit of knowledge about how membranes work. This is, we said siRNA. This is a little bit of, again, some molecular biology knowledge of RNA types. And this is our only question that it was about experimental analysis, right? And this one, it seems to be that from our interpretation of the graph, donor livers have higher levels of CX43 after reperfusion when compared to normal liver tissue. So we see that choice A is correct. It's a connexin protein, and connexins form gap junctions. Number 2, C is correct. The inhibitor described in the question stem is very hydrophobic, and this means it will pass through the membrane via diffusion. So this is 2 pieces of knowledge. Number 1, do you know that triglycerides are hydrophobic, or can you determine that they are? And number 2, do you know how hydrophobic stuff passes through a membrane? Does it pass through a channel, or does it pass through directly through diffusion? In which case, it's diffusion, right? Answer choice A is correct. siRNAs bind to mRNAs for certain genes and lead to degradation. If the RIP1 mRNA is degraded, CX43 will no longer be able to interact with RIP1, right? So we talked about how the other ones don't make sense. The increase in CX expression should not disrupt the interaction, right? It would increase it. Answer choice C is correct. The graph in figure 1 shows that donor livers have higher expression levels of CX43 mRNA when compared to normal liver tissue. The mRNA expression levels given by the graph and the protein expression levels given by the blot are different, meaning that regulation occurs at both the transcriptional and translational levels. Oh, okay. And then heptanol is not addressed in figure 1, like we said. So they said review transcription versus translation, practice interpreting experimental data, right? This was the only one that actually made us practice experimental data. So the whole point is, when you read a passage like this, don't get freaked out. Even if you think like, oh my God, I have no idea what I read. What are they talking about, right? You don't need to necessarily know everything of what they're talking about. You need to be able, what you need to do is pick out the keywords, the key sentences that allow you to answer certain questions based on your content knowledge, right? Because look at number 1. Number 1 was saying, what type of junction does CX43 most likely form? The only thing that you need from this passage is to look at what CX43 actually does and then connect it to your own knowledge of what a gap junction is, right? So when you read a passage like this, do not get intimidated. Think about, let me take it one question at a time and see what they actually are asking me about. Most of the time, there's only 1 or 2 questions asking you to understand an experimental part. And again, they're usually not asking you to understand the entire experiment. They're usually just asking you to understand maybe one graph or one aspect of the experiment. So yeah, so that's a bio passage, and I think that's it for today. So let's see.